Let's talk about species today. So, first off, define species, speciation, microevolution, macroevolution, allopatric, sympatric, autopolyploidy, and allopolyploidy. Be able to list and define the different species concepts. Compare microevolution and macroevolution. List methods of pre- and post-zygotic isolation. List reproductive barriers and give examples. And compare allopatric and sympatric speciation. What is speciation? Well, it's the formation of new species. There are species concepts we're going to look at that define what a species is and when a species is distinct. And we should know that the rate of speciation differs between different groups. You're going to see that a lot. A species is a set of related organisms that maintain a certain level of traits in nature. We can also look at subspecies and ecotypes. So, what determines the species designation? What is a species versus what is a genus or a family? Well, a species can be identified 4 different ways. We can look at the morphological structures, i.e., what does this organism look like and does it look like different things? We can look at reproductive isolation. Does this organism breed with others that are similar looking? Or molecular features where you can actually look at the DNA and say that this organism is distinct via different DNA sequences, or ecological features where this organism occupies a different niche than others. There are multiple different species concepts, and we're going to look at the pros and cons of each one, starting off with the morphological species concept. Basically, if it looks like a duck and walks like a duck, it's a duck. This means that we're looking at the physical characteristics. A problem with that is our looking at physical characteristics is not how other organisms look at different species. What they do is they may look at the scent, or we may have sexual dimorphism where the male and the female actually look very different, but they know darn well that they are the same species. So there is a positive to this and a negative to this. Another positive is this works really well with fossils. The ecological species concept, we're looking at what resources they use in nature. And this tells us basically where they are in a niche. And we can look at things like sticklebacks, where there are limnetic sticklebacks, which live closer to the surface of ponds, and benthic sticklebacks, which live down in the depths of ponds. These organisms definitely recognize each other as different species because they occupy different ecological niches. The phylogenetic species concept is when we look at the different DNA. This will say that if 2 populations have been isolated long enough, they're going to have different DNA sequences, and that is an easy way to tell if they are different species because they haven't been interbreeding. But all of this looks at interbreeding a lot, which is why we're going to focus on the biological species concept, which looks at reproductive isolation. There may be a lot of morphological differences within a species. They may occupy slightly different niches. They may be phylogenetically different because they've been isolated for a long time. But if these are 2 organisms that would get together and mate and produce viable, fertile offspring, the biological species concept will recognize that morphologically they will become more similar as they share alleles. Ecologically, they will become more similar if they're going to be interbreeding and therefore occupying a similar niche when they are interbreeding. And phylogenetically, they're going to become more related if there is gene flow between populations. And gene flow is really where the biological species concept parks itself. So let's look at microevolution, which is change in frequency of traits in a population. We've been looking at that quite a lot, and that can lead to reproductive isolation, causing barriers that impede individuals from interbreeding and thus sharing genes. So this is going to stop gene flow via reproductive isolation, and over time, that is going to cause alleles to go to fixation, where the different populations have distinct alleles. So what does that cause? Well, let's see, we have 2 populations here that are interbreeding and sharing different alleles and thus have the same traits. But something is going to come between them that is going to make these populations stop interbreeding as much. It might be natural selection, sexual selection, genetic drift, or some mutations that cause them to not interbreed. And when that happens, we are going to see divergence. This means these 2 populations are going to become less and less like one another, causing them to become morphologically distinct, ecologically distinct, phylogenetically distinct, and thus they are biologically different species. And this is where microevolution becomes macroevolution, the formation of new species. And we know they're different species when they're reproductively isolated, so let's look at reproductive isolating mechanisms. There will be prezygotic and postzygotic isolating mechanisms. Prezygotic means the zygote is not formed. So that first arrow down there, that means something's going to happen before that first arrow occurs. Postzygotic is anything after the arrow, where a zygote is formed and then something goes wrong. So let's list them out. There may be more than one. They may not be complete, and this may cause hybrids to be formed. So do understand, this is not a one-size-fits-each example, but rather multiple different things may be going on here. So first off, let's look at habitat isolation, or some sort of geographic barrier that is going to prevent contact. If you have two snake species here that are going to dwell in the water or terrestrially, they're not going to mate with one another because they are in different habitats, even though those habitats may be close to one another. Or lions and tigers live in different habitats. Lions being primarily African and in the Middle East, or formerly in the Middle East, and tigers being primarily in the farther east, such as India and Sumatra and the Amur Basin. So these species do not come in contact with each other in nature. However, they can come in contact with one another in captivity, and if they mate in captivity, they can be forced to make hybrids. So as we see, some of these barriers may not be complete, and we have to consider what happens in nature for the biological species concept. Temporally, they could reproduce at different times, either different times of the day or different times of the year. An example of two toad species, if they are mating at different times of the year, then they are not going to have their gametes come in contact with one another and make zygotes. Likewise, with the skunks, they may not come in contact with one another, and they may not even be in season. If one meets another and one is feeling like mating, the other one may not be ready to mate at that time. Behaviorally, organisms can distinguish between different species based on their behavioral traits. So these two meadowlarks may have very different calls and thus be completely unattractive to one another because they are different species. Blue-footed boobies have a mating ritual where they dance around and show their feet off, and the moment one of those feet is red, such as the red-footed booby, that is not attractive to the blue-footed booby. Pheromones play a strong role in this as well, with different butterfly species smelling different and thus being behaviorally attractive only to those of the same species. Mechanically, there may be morphological features that just prevent successful mating. Like with his Bradybaena nails, what happens is their mating apertures are on the opposite sides, so they cannot mate because they may try as hard as they can, but they simply cannot line up their reproductive apertures. Or damselflies, where they need to be the same length of abdomen in order to actually do their mating ritual. The gametes may simply not fuse. So sea urchins have a tendency to just release their gametes into the water, and if they do not fuse, they cannot make a zygote. Trinidad guppies, although they may try to mate, the male's sperm simply do not unite with the female's egg if the males are from different streams, making them gametically isolated. But if those gametes get to unite, they will be fertilized, and that will cause a hybrid. So let's look at postzygotic isolating mechanisms. Starting with hybrid inviability. The eggs are fertilized, but something goes on with the embryo that the embryo is not going to survive in utero, and it will just be aborted or will not survive in the real world, such as this Ensatina hybrid, which one parent lives in moister environments, the other parent lives in drier environments. This one can't live in the in-between environment because the in-between environment simply doesn't exist where they are formed. Sterility may occur where the hybrid may be perfectly livable, may survive to a ripe old age, and may be completely sterile like this mule here. The offspring of a donkey and a horse can live quite a long life, but will not ever produce offspring. It just doesn't have the right gamete-making abilities. They could make gametes, but those gametes may unite and form inviable offspring or less and less viable offspring as the generations go on. This is known as hybrid breakdown. The hybrids are viable, but subsequent generations have reduced fitness over time. As there are more rainbow trout alleles in the cutthroat trout population, the mean number of offspring per cutthroat trout female decreases and their fitness decreases. These reproductive isolating barriers maintain the adaptations specific to each species, so it is beneficial for a grizzly bear to only mate with other grizzly bears and a polar bear to only mate with other polar bears if the hybrid would not be as strong in the environment. That means that their offspring would be less fit, so it benefits the fitness of the adults to only mate with other adults of that species. And this will maintain those isolating barriers and maintain species integrity. Let's look at the mechanisms of speciation. We will start with allopatric speciation, where a geographic barrier is going to impede gene flow between populations, allowing speciation to occur. Say there is a pond here, and that pond dries out over the long term, which is going to cause these two fish species to be unable to mate with one another. There's no gene flow. Later on, the pond forms again, and the two species are different species, and they recognize each other as such, thus allowing for more isolating mechanisms to evolve. Canyon is a good example here. The chipmunks on each side of the Grand Canyon cannot mate with one another, therefore it forms a barrier to gene flow, and they have speciated over time such that they now mate at different seasons as would be appropriate on each side of the canyon where slightly different ecological features exist. It is also possible that there is reproductive isolation because of different body forms, such as the different body forms of these guppies under high predation or low predation. As these body forms differ, so we will see prezygotic isolating mechanisms, either mechanical or behavioral, as they are either unable to mate with one another or behaviorally isolated because they look very different. Another example of this allopatric speciation is in the species of snapping shrimp that are formed on different sides of the Isthmus of Panama, which rose 5 million years ago. And as it rose, you can actually see the different species of shrimp forming. The deepest species formed first because they were isolated first, and the most shallow species formed last because they were isolated last. We see an example of allopatric speciation in refugia, where a once contiguous rainforest has been fragmented into smaller little zones where the dusky salamander can survive. In each of these little zones, they are isolated from other populations of dusky salamanders, and it is possible that over time, new species of salamanders may form in each of these refugia. We also can see this when populations move to new geographic regions. The Eurasian rosefinch had a population blown away, think of this founder effect, to Hawaii, where they formed different species as they evolved into new niches with new adaptations. Now, allopatric speciation, remember that the physical barrier initially reproductively isolated populations. Populations diverged, and over time they evolved into their own niches with their own behaviors and their own body shapes, causing prezygotic mechanisms to evolve. When those populations are re-exposed to one another, you will see these isolating mechanisms strengthened to ensure species integrity. There is also sympatric speciation, where there's no geographical barrier and there's nothing impeding the gene flow between populations, but they still speciate. How can that happen? Well, several ways. First off would be polyploidy. A genetic change can immediately cause reproductive isolation. What does this look like? Well, in certain angiosperms or ferns, you'll actually see what's called autopolyploidy, where an individual with two chromosome sets is going to be derived from a single species. There's a cell division error in meiosis, and there's going to be a tetraploid cell produced as an offspring. Plants are able to fertilize themselves quite well, and that can form a new species that is then not viable with the original diploid species. Allopolyploidy is when there's a hybrid with two chromosome sets from different species, where pollen from one species lands on the ovule of another species and they form an offspring that is a hybrid. This hybrid then either mates with the original or mates again with itself, causing a viable fertile hybrid. And this can happen when there is just the pollen overlap between different species of plants, and of course is prevented when they are temporally isolated, when their pollen doesn't come out at the same time of year. Habitat differentiation also causes this. The hawthorn tree is grown alongside apple trees, and flies can move between hawthorn trees and apple trees, but they prefer to mate and produce offspring on their own trees. When they mate and produce offspring at their own trees, they have to actually lay their eggs at different times of year, causing temporal isolation to arise between these two species of flies. We can see this on different aphids that live on alfalfa and clover, or different walking sticks that have different ecotypes based on the different species of plants on which they live, and thus the camouflage they have to have. So these little adaptations cause the intermediate form to actually be less viable than either of the species, and thus reinforces the isolating mechanisms. Sexual selection can also occur with these fish species that have very distinct mating patterns. These mating patterns are enhanced in shallow or deeper water because of how deep light penetrates, such that certain mating patterns are more attractive in shallow water species and other mating patterns are more attractive in lower waters. Now, if you just give them orange light, which gets rid of the blue light and the red light, and then have them in a common tank, they can't tell the difference. But in their native habitat, they can tell the difference. They do tell the difference, and they are speciating. We see this in finches as well with different types of calls. The mating calls are differentiated, and thus they find each other less attractive if there is a different species making that call. Remember, both allopatric and sympatric require an agent of microevolution and reproductive isolation. As reproductive isolation goes forward, there is that geographic barrier, or there is that mating barrier, and more reproductively isolating barriers may evolve as a byproduct. So sympatric speciation requires emergence of that reproductive isolation without a geographic barrier, and we can see these being reinforced.